Well, good morning. My name's Joel. I'm a PhD student at Swansea University. I'm studying under Professor Juan Mareke Rivas. And we've been looking at the uses of diatoms in immunotherapy and cancer treatment. So just to give you a bit of background into diatoms then, they're naturally found in bodies of water where there is sufficient light and nutrients. They're a unicellular microorganism encased in a biosilica shell. They're responsible for about 20 to 40% of photosynthesis on Earth, comprised of two valves loosely joined together. If you look at this image here, you can see the porous valve structure there, and then there are very loose structures which join this valve with the valve at the other end as well. They're not directly linked, but they're sort of slotted into one another. If you see here as well, this should give you a better representation. When the diatom reproduces, the microorganism inside grows, which pulls the two ends apart. The microorganism then splits, and then new valves and new structures in between then form to create two diatoms from the one. They've currently found uses in a range of industries, including photonic crystals sensing, biosensing, filtration, separation, and catalysis. And they're also being heavily used in biomedicine and drug delivery as well. And that's the work that we've been carrying out here. So just to give you a bit of an idea of the concepts that we've been trying to develop. So we've predominantly been looking at loading these nanoparticles with loading these diatoms, with different nanoparticles, different small molecule therapeutics and DNA and enzymes as well, to help initiate a variety of different anti-cancer responses. So we have a loaded diatom here. The concept is that we can then implant this into a localized tumor cell. And then the therapeutics that have been loaded onto the diatom can slowly be released into the environment and initiate the anti-cancer responses locally. So one way we've been doing this from an immunotherapeutic perspective is we've attached double-stranded RNA to the diatoms. These are then internalized into dendritic cells, which are picked up by toll-like receptors. These then mature the dendritic cells, which then go on to activate T cells, and then the activated T cells can then go on to initiate an immune response against the cancer cells. So we've been wanting to carry out this work with three different species of diatoms. We have Thalassiothera weissflogi here, Thalassiothera pseudonana, and Chytoceros calcitrans. We've predominantly been working with Thalassiothera weissflogi. It's the one we've been able to culture the greatest quantities of. And then once those concepts are finalized and optimized, we'll then go on to the other species as well. So in terms of loading different therapies onto the different therapeutics onto the diatoms, because of the way we've treated the diatoms to purify the different biosilica structures, it involved a wash with hydrogen peroxide, and this activated some hydroxyl functional groups on the surface of the diatom. So this then gave the surface a partial negative charge. Now, we've been working with some polyethylenamine-coated gold nanoparticles, which are positively charged, which will directly attract to the negatively charged diatoms. And then we've also been working with poly-IC and imiquimod, which are immunotherapeutic molecules. They are toll-like receptor agonists, so they can help initiate an immune response as well. So we've been trying to do sort of multi-layer loading. So we first began by attaching the gold nanoparticles onto the diatoms, and then from there we went on to attach the poly-IC with the thinking that the negatively charged molecule will attach to the positively charged molecule. This thinking turned out to be correct. So we characterized the diatoms before and after gold nanoparticle attachment using scanning electron microscopy. You can clearly see after the attachment the white dots, which represent the gold nanoparticles. So this was indicative of a successful attachment of nanoparticles onto the diatoms. And then we looked at loading the different immunotherapeutic molecules, poly-IC and imiquimod, onto the diatoms as well. We attempted it with no gold nanoparticles, and there's a very slight attachment here to one of the species, but we put that down to instrument error. Ultimately, there was no attachment, which we expected. And then when we attempted to attach to diatoms with gold nanoparticles, we could see that there was significantly more attachment in both poly-IC and imiquimod as well. So another way we've been, or another angle we've been attempting with anti-cancer treatment is to utilize the intrinsic peroxidase activity of iron oxide nanoparticles, and to also see if there was intrinsic peroxidase activity associated with the diatoms as well. So standard biochemistry in cells, glucose will react with oxygen in the presence of glucose oxidase to produce gluconic acid and hydrogen peroxide. Now, iron oxide nanoparticles will undergo Fenton reactions and react with hydrogen peroxide to produce free radicals or reactive oxygen species, and these can then go on to initiate oxidative stress in cancer cells and then go on to initiate cell death as well. This has a dual approach in the sense that it will, sorry, going back from there, we then attached, so under this concept, we then attached iron oxide nanoparticles to the diatoms using electrostatic interactions again. We then also attempted to attach glucose oxidase to the iron oxide nanoparticle-coated diatoms with the intention of promoting or further promoting the biochemical reactions in the cells. So this had the dual approach of producing the reactive oxygen species, which are damaging to the cancer cells, but also then used up the glucose supply, which starved the cancer cells of their food source. So it had a dual therapeutic approach, and was a nice proof of concept for combination therapy as well. So we were able to characterize the catalytic activity of our systems using a colorimetric substrate called TMB. So in the presence of the free radicals, TMB is oxidized to this charged substrate here. It produces a nice blue color, which is detectable at 652 nanometers by UV vis. So we observed the change in absorbance over time at different concentrations of hydrogen peroxide. And we were able to see that the higher the concentration of hydrogen peroxide, the greater the catalytic activity response. And from that, we would then be able to go on to calculate kinetic parameters and then be able to compare that with other systems that have already been characterized for their catalytic activity. Another angle then, we also went on to carry out a surface modification of diatoms with the intention of attaching an antibody for immunotherapeutic purposes and for detection purposes as well. So this surface modification here, we attached aptes to the surface of the diatom. This has already been published, so we carried this out and then took this further and went and attached sulfo-SMC, which is a cross-linker to the amino groups on the aptes. And then from there, we then went to attach an antibody onto the cross-linker. We don't believe the antibody attachment has ever been carried out before. But surface modification of diatoms is currently well documented. So from an immunotherapeutic point of view, tumor cells are very good at avoiding the immune system by expressing program death ligand 1. T cells are then able to detect that ligand using program death 1, and that then prevents an immune response. Using checkpoint inhibition, we are able to use this antibody to block the program death ligand 1 molecule, which prevents the T cells from registering it, which then does not prevent an immune response. So the T cells then go on to attack the cancer cells. It also has the potential to detect various molecules or various ligands as well. So A system that we've been trying to work with is we have this antibody modified diatom. We also have some antibody modified nanoparticles, and we've attempted to capture the nanoparticles with the diatoms using programmed F ligand 1. And then measuring the difference in the amount of nanoparticles left after the attachment, and that would then be indicative of whether or not somebody is eligible for a certain type of immunotherapeutic treatment. It's ongoing work, but we're very hopeful of a concept that we can then go on to publish. So just to conclude then, we've demonstrated that the diatom's ability to capture nanoparticles we've demonstrated that the diatoms do have the ability to capture nanoparticles and small molecule therapeutics in very simple and very straightforward chemistry. The peroxidase activity of these systems has also been demonstrated and shows great therapeutic promise. The attachment of antibodies to the surface of the diatom has also been carried out as well, which also shows great promise for future applications. Going forward, we want to carry out more in-depth in vivo studies into the various diatom systems with nanoparticles and small molecule therapeutics, and to look at the catalytic activity in vivo as well. The release of nanoparticles and small molecules from the diatoms has not yet been characterized. That's something we also want to look at. And then also exploring further the various uses for antibody-modified diatoms. So I'd just like to acknowledge my supervisor, Professor Juan Mareke Rivas, who's been very supportive throughout and is on his recommendation I'm now doing a PhD. Saul, who spoke this morning, was very helpful in the early days of my studies as well, as was Mark, who I believe is speaking tomorrow. He was also very helpful. So big thanks to all three of them. That's it from me. If you have any questions, I'll do my best.
Thank you very much, Joel. A beautifully clear talk. If I may just start with the first question, because these are so ubiquitous in the environment, we must ingest a lot of them.
Yeah, so we when we initially started this work, we bought some commercially processed items, and they were designed for athletes as a food supplement. So yes, they can be ingested.
And do they show benefits in athletes? Is that why they take them?
They're designed as silica substitutes. I'm not 100% sure on the exact benefits, but there does appear to be a market out there for them.
It must be based on sound science.
Yeah.
So I've got a question from Dr. Neil Liptrot. Hi, Joel, fascinating talk. There are known adverse reactions to immune checkpoint inhibitors, such as skin reactions, even though they're administered systemically. Do you see similar issues here?
So we haven't got to that point in the studies where we've been able to characterize those types of adverse reactions. The main focus of the work with checkpoint inhibitors has been looking at the sensing applications. So that's where the main focus of the work has been so far. But I do believe we are hoping to go on to look at sort of the various delivery of checkpoint inhibitors and whether a concept can be produced where it can be delivered locally, so those adverse reactions can hopefully be minimized.
Thank you. And so a question here from Nugent Than. Do you know the efficiency of conjugating antibodies? Which assay did you use? Do antibodies keep their activities after conjugation?
So we've had a range of efficiencies. For the reactions that we've used, we've managed to attach around sort of 30 to 40% of the antibody that we introduce into the reaction, and that equates to about 30 to 40 micrograms per milliliter in reaction in a solution that contains 1 milligram of diatom, I believe. We use a BCA assay to characterize the attachment. Was that all of the questions? Sorry.
That's all. May I just jump in with one final one? There must be a lot of natural variation, again, because they're so ubiquitous. So depending on where you are in the world, maybe, are they influenced by climate?
In terms of
their structure, their size?
Yeah, so I mean, you know, we haven't we haven't looked into every single type of diatom species, but yes, naturally there are a huge amount of diatoms. But one of the attractions or one of the appeal is that they can be grown, or a single species can be grown very simply in the lab. I could go to the biology department at Swansea University tomorrow and ask for a 20-litre sample, and they'd have it ready for me in 2 to 4 weeks. So we've been able to work with single species there. I don't see that there would be a need to harvest them from natural resources off the back of that, but yeah.
We'll leave it there. Maybe I'll follow up with my rest of my questions. Thank you, Joe.